Hello everybody, greetings of the day and welcome back to my YouTube channel. And today we are going to discuss about the tips and tricks to crack GATE examinations. GATE Excel Life Sciences, that is our main topic for today. So this video, if you follow few tips and tricks very with being very focused and disciplined, for sure you can crack GATE in your first attempt itself. So target GATE is our main motto for today. So myself Payambora and I'm GATE qualified 2022-2023. So today I'm going to discuss a few of my experiences with you all that may help you all to solve the GATE paper or to score a very good mark. So GATE 2024, it is a computer based test. So that means it is an online test. The duration is of 3 hours. The number of subjects, there are 30 subjects, but today we are going to discuss about the Excel code, life sciences. So there are two sections basically. One is your general aptitude, where your verbal reasoning, your aptitude, all those general knowledge questions appear, grammars, mathematics. And then the second question is your domain specific, that is your selected subject. So here you have food technology, microbiology, biochemistry, zoology. These are few of the selected subjects where you can attempt. So that we'll be discussing ahead. What are the types of questions? So there are MCQs, that is multiple choice questions. There are MSQs, multiple select questions. So there can be two or more right answers for that one question. So you will get the answer, you will get the correct mark only if you answer all the correct options. So that is one twist here. And numerical answer type. So you will be given an empty column, and suppose if the answer is 2.18 into 10 raised to 5 or something like that. So you have to type that answer. If it matches, then you will be up to one digit, up to 1 point after decimal, then you will be awarded as 1 mark for that question. So that is how there are total 65 questions for 100 marks. There are total 65 questions for 100 marks, out of which your 10 question is your general aptitude one, general knowledge one, and your remaining 55 questions will be your domain specific questions. So what is the main thing that you need to remember? Just type GATE Excel Life Sciences previous year question papers. So once you type these, you will get a number of questions papers over here. You can see the site over here, and there are so many different sites where you can see there are previous year questions. So solving PYQs can help you get more acquaintance upon the types of topic, the most repeated questions, so that you can easily choose those topics. So that is why these topics are very important, previous year questions. Next up, what will be your target? Now, if you want to crack your GATE, your domain specific questions must work. So your chemistry, your zoology, your biochemistry, your microbiology, your food technology. These are the 4 domains from which you have to select 3. You cannot opt for all 4. So from zoology, what is your, if you are strong in zoology, you can choose this. If you are strong in carbohydrate, protein, lipids, vitamins, and all, you can opt for this. If you are strong in staining, if you are strong in culture, assay, if you are strong in biotechnology, something, and if you are strong in food technology, so you can opt for these topics. I had chosen zoology and food technology. The 2 topics that I had chosen were food technology and zoology, and there you get a chance. So in the, there is no as such necessary that in your form that you're filling you have to attempt or you have to write any or prioritize. Whenever you're giving your exam, at that time you can check all the 4 papers. So you can check what are the questions of zoology, biochem, microbiology, and then you can select accordingly, okay, how much questions you know from this domain, and then you can choose to any 2 domains that you want to opt for. So high weightage questions, there are certain questions that are high weightage. You cannot leave them. It is a compulsory for you to know those questions. One is organic name reactions, your basic 12th reactions like Grignard, Wolff-Kishner, Clemenson, Sandmeyer's. Those questions are quite frequently asked. Reaction kinetics. So you have first order, second order, zero order. You need to know the half-life, shelf life, the units. Chemical equilibration. There are questions asked from chemical equilibration from your standard 12th. SAR structure related questions are quite asked. Biomolecules, of course, there has to be a question on biomolecules, macromolecules, micromolecules, carbs, protein, lipids. So you have to be aware upon the cycles, glycolysis, Krebs cycle, whatever, all the cycles you must know, the intermediates, the enzyme converting, the number of ATPs required and the number of ATPs formed, your electrochemistry, so your cells, your electrode, primary reference electrode, secondary reference electrode. Thermodynamics, you need to know the laws of thermodynamics, the zeroth law. There was a question in GATE 2022 on zeroth law. So you need to know the laws of thermodynamics. Atomic structure, so you need to know that electronegativity order will reduce down the table or across the table. How is it? The acidity, the basicity. So you need to know the this kind of relationships, that if there is high electronegativity, the acidity is more. So you need to know these terms. So that is why these are of high weightage tips. Now speaking about the syllabus, so syllabus has your chemistry as your first part. So this is a big syllabus. To be honest, covering so many topics is genuinely very difficult, especially for a GPAD aspirant or for a pharmacy student, or for any, if you're microbiology, biochemistry, whichever domain you are. But covering so many topics is very difficult for you. So there are few topics that you cannot leave, like VSEPR theory, valence shell electron theory. That is important. Here you need to know the ionization energy, the trends, ionization energy, electronegativity, the atomic size, which is fluorine, chlorine, bromine, iodine, which is amongst the halogen, which has a higher atomic size. That is important. Now, if we speak about chemical equilibration, you need to know about atomic, you need to know about osmotic pressure and pH and buffer. So these are colligative properties. These are three topics that you cannot leave: colligative properties, pH, buffer, and osmotic pressure. Speaking about electrochemistry, so you need to know the EMF series, electromotive forces series, Nernst equation, which is very important. The numericals are asked upon Nernst equation, and the Colerache law. So that is a cell law. In the reaction kinetics, I already mentioned, you need to know the order of reaction, the difference between order and molecularity. So that difference is very, very important. You need to know the difference between order and molecularity. Next, thermodynamics. So you need to know the Fick's law, the three laws of thermodynamics, which law deals with entropy, which law deals with energy, activation energy and all. So you need to know that. Next is your section 8, from where you need to know a lot of topics like isomerism, stereochemistry, SN1, SN2, your name reactions, your functional group identification tests. Here you can see, so these are topics that you cannot leave. These are must topics. Speaking about the section 9, so in the section 9 you have biomolecules. Again, all of this is to be done, Beer-Lambert's law, which is in your semester 7, your UV spectroscopy, A is equal to ABC. Carbs, proteins, lipids, their identification tests, their classification, everything is to be done. They focus more on ion exchange and gel filtration. So see to it that you know the principles and the names of reagent used. Like in ion exchange, we have cationic exchanger, we have anionic exchanger, we have strong cationic, weak cationic, strong anionic, weak anionic. So you need to know the examples, because this was a previous year question. Let me tell you, this was a previous year question. Gel filtration, so it is based upon molecular weight and molecular size. So that is why this is very important. Protein sequencing. There are different methods for peptide sequencing, like Edman's method. You have phenyl isothiocyanate in that, Sanger method. So there are certain methods for sequencing that you cannot leave. Next up, we have biochemistry. So biochemistry mein you need to know the enzymes and their functions, especially hemoglobin, chymotrypsin. These are few enzymes. You need to know the application in industry as well. Like chymotrypsin is used for cheese as well. So you need to know this, where that's very important. Section 2 may we have enzyme kinetics, we have vitamins and coenzymes. So there is a list of 8 coenzymes and their particular cofactors. Like suppose if we have selenium, so glutathione peroxidase is a is an enzyme by that is governed by that cofactor. So you need to know that. The cycles, as I said, you cannot leave urea, Krebs, citric acid, shunt cycle at all. At all, it is to be done. The rings in the vitamins, the deficiency of vitamins, the synonyms of vitamins, those are to be done very well. In your section 3, you have protein sequencing method, same that you covered in biochemistry, that you covered previously. So you need to know that. Spectroscopy mein a little knowledge about quenching fluorescence will do no harm. Cell structure, yes. So prokaryotes and eukaryotes, you need to know which are the organelles that are cell membrane bound, which are double membrane bound. So those questions are quite frequently asked. The types of receptors, the 4 types of receptors: ligand gated, G protein, ion linked, enzyme linked, and nuclear. Next up we have is transcription translation, your central dogma: replication, transcription, translation. You need to know all the enzymes involved, all the things involved, like Okazaki fragments. We have DNA ligase, we have RNA polymerase, DNA polymerase, RNA primase, so many different enzymes involved. So you need to know topoisomerase. So enzymes and their functions, that is suffice. Section 6 is immunology. So immunology and serological diagnostic test is a must. There is a table that is available of diagnostic test, the disease and the diagnostic test. So it is to be done. And next up, if we say the structure of an antibody, Y-shaped antibody, MHC complex, major histocompatibility complex, MHC 1 and 2 ka difference is asked quite often. Production of monoclonal antibodies, so you should know the media. PEG is used, polyethylene glycol. Then salvage pathway is important. HGPRT is the enzyme. So these are few short things that are, we can say these things are only us, nothing else, nothing theory. All your concept based. ELISA, so direct, indirect. ELISA में which enzymes, horseradish peroxidase, HRP is used. So these are the things that are very frequently asked. So you need to know them very well, thoroughly. PCR, you cannot leave PCR, polymerase chain reaction. That is very important. And the rDNA, the tools of rDNA, vectors, the plasmids, cosmids, bacteriophage, artificial chromosomes, the restriction endonuclease, the types of REN, RAN, REN, restriction endonuclease 1, 2, 3. So you need to know them as well. Next up, we speak about botany. Botany is, to be honest, for pharmacy students, it is a big no, that don't go for botany because it is difficult, because there are miRNA, siRNA, euploidy, aneuploidy, the topics that are asked. The C3, C4 pathway, that is the Calvin pathway. Then you have various plant hormones like auxin, cytokines, gibberellins, abscisic acid, ethylene, on which question are asked, and that anatomy, morphology especially and all. So if you are not good at your in your botany, I would suggest leave it, because these topics are usually not covered in your pharmacy. These are some miscellaneous topics like ecology. May you have consumers, primary consumers, producers, herbivores, carnivores, all those things you might forget. So it's better that you not get seer. You have Krellox pathway, you have CRISPR pathway. These are difficult. You need to learn them. Chimeras. So these are chimeric antibodies. These are very difficult to remember, to be honest. So I would suggest opt for microbiology, biochem, or food tech. Any two of them, but don't go for such topics. In microbiology, I would suggest PCR again, as I mentioned, and electron microscopy. So microbiology means you cannot leave microscopy. You need to know simple microscope, compound microscope, the uses, the applications, like which one is used for unstained, like phase contrast microscopy is used for dead or living. So that you need to know. As I mentioned, prokaryotes and eukaryotes, entire structure, like you need to know the bacteria, capsule, cell wall, cell membrane, the synonymous names for them as well. Metabolism, you need to know the citric acid cycle, C3 cycle somewhat, a little glimpse of those cycle. Embdon-Meyerov pathway, which is glycolysis. Diseases, what are the opportunity disease, what are the hospital acquired disease, what are nosocomial disease, what are those disease that are, you need to know the microorganisms. Like if I say Salmonella typhi, so you should know immediately that typhoid is caused by. If I say Yersinia pestis, so you should know that it is causing plague. If I say Clostridium botulinum, so you should know that it causes food poisoning. So these are few of the microorganisms and the disease that are caused by these microorganisms. If I speak about vaccines, so vaccines may you need to know the classification, which is live, attenuated, which is killed vaccine, again which is viral, which is bacterial. The classification of vaccines is extremely essential for you to be studied. So I would suggest that please study vaccines अच्छे से. Next up, we have chemotherapy or antibiotics. So chemotherapy and antibiotics is a must. Guarantee question from chemotherapy and antibiotics. So you have to do everything about antibiotics, the mechanism of action. In 2022 there were 2 questions, in 2023 there were 2 questions. So you need to know the assay, which microorganism is used for which antibiotic assay, then which antibiotic is causing its mechanism of action, what is acting on 30S, 50S, cell wall, cell membrane, everything. You cannot leave anything from here. If you speak about genetics, so you need to know about transposons, which are jumping genes, Lac-Operon model, lambda phage, the vector capacity, like there is a particular capacity of how many kilo base pairs, KBP we call them. What is transduction, what is conjugation, what is there was a question on conjugation as well. Ames test, which is a test for carcinogenicity, mutation. So you need to know this very well. Next, zoology. Zoology may usually have you genetic basis of inheritance, you have sexual diseases, you have like those topics of telomere, chromomere, centromere, all those things. Biochemistry somewhat is included, like nucleic acid, protein, vitamins, lipids, amino acids, gene expression pathways. You have cell division, mitosis and meiosis involved. Animal anatomy and physiology. So you have skeletal system, endocrine system, all your HAP, all your semester 1 pharmacy HAP part. Parasitology and immunology, you have immune system again, immunology, opsonization, precipitation, agglutination. There are few terminologies that needs to be at the tip of your tongue and their definition. The last subject is food technology, which is has a code of XLU. So food technology is very scoring subject. Trust me. You have enzymes, you have nutraceuticals, you have spirulina and all covered. You have cofactors, you have nutrients, which are macronutrients, micronutrients. You have polymorphism, you have rancidity. You have those tests: acid value, saponification value, iodine value on. Food microbiology, you have staining, gram staining, you have streak plate, pore plate method for staining. Then you have to like toxins that are formed from microbes, and like what are the fermented beverages. The fermentation is a very important topic. You cannot leave. There are two terminologies of fermentation: trophophase and idiophase, which are used for primary and secondary metabolites. Like tropho is for primaries, idio is for secondary. Then you have upstream and downstream. So which one is for recovery? You need to know that very well. So that is why this food technology is very scoring. I must say I had attempted food technology only. In food engineering, you have which food, like you have so many citric acid, citrum, you have cornybacterium, glutamicum, you have micro aspergillus niger. So these kind of questions directly they ask that which is the organism for fermentation. Like acetobacter acetize used for vinegar. So they directly ask this question. It was the last year question. You have Reynolds number. So from pharmaceutical engineering, there are few evaporators, crystallizers, humidifiers, drying machine, evaporators, filtration. All this you have covered in pharmaceutical engineering semester 3. Next up, we have processing principles. So in processing principles, usually it is difficult to remember, trust me, because HACCP, hazard analysis and critical control points, clean in place. This is all QA part. So QA, QC, you can study well. So that's all for today's video. I hope you all did enjoy and did get an idea about GATE, the topics of GATE, and which are to be more emphasized upon. So that's all for today's video. If you have any doubts, please do feel free to drop them in the comment section. I'll be more than happy to answer it. Thank you so much, everybody.